Hello, welcome to Biology, the scientific study of life. Many human traits follow Mendelian patterns of inheritance, whereas bees are convenient subjects of genetics research. Humans are not. The human generation span is about 20 years, and the human parents produce relatively few offspring compared to bees and most other species. Furthermore, breeding experiments like the one Mendel performed are unacceptable to humans. In spite of these difficulties, the study of human genetics continues to advance, spurred on by the desire to understand our own inheritance. Now, new technique in molecular biology have led to many breakthrough discoveries. But basic Mendelism endures as the foundation of a human genetics. Pedigree analysis, unable to manipulate the meeting patterns of people, geneticists must analyze the results of meetings that have already occurred. Notice they do so by collecting information about a family's history for a particular trace and assembling this information into a family tree describing the interrelationships of parents and children across the generations, the family pedigree. Notice this diagram shows pedigree analyze this family trees squares represents males and cycles represents females. This diagram. A horizontal line connecting a male and female like this indicates a mating with offspring listed below in their order of birth from left to right. Shaped squares and circles represent individuals who accept the trait being traced. Dominant trait windows beak. Windows beak, this pedigree traces the trait called windows beak through 3 generations of a family. Notice in a 3rd generation that the second born daughter lacks a windows beak. This one, inheritance supports although both of her parents had the trait, such a pattern of inheritance supports the hypothesis that the trait is due to a dominant allele. If the trait were due to a recessive allele and both parents had the recessive phenotype, then all of their offspring would also have the recessive phenotype. Another notice also in this diagram recessive trace attached aerolop. Notice this one. This is the same family, but in this case we are tracing the inheritance of a recessive trait attached aerolops. Notice that the firstborn daughter in the 3rd generation has attached aerolops, although both of her parents lack that trait. They have 3 aerolops. Such a pattern is easily explained if the attached loops phenotype is due to a recessive allele. If it were due to a dominant allele, then at least one parent would also have had the trait. Recessively inherited disorders. Thousands of genetic disorders are known to be inherited as simple recessive traits. These disorders range in severity from relatively mild, such as albinism, lack of pigmentation, which results in susceptibility to skin cancers, and vision problems, to life-threatening, such as cystic fibrosis. We account for the recessive behavior of the alleles causing these disorders. Recall that genes code for proteins of a specific function. An allele that causes a genetic disorder coded either for a malfunctional protein or for no protein at all. In this case, or in the case of disorders classified as recessive, heterozygotes are normal in phenotype because one copy of the normal allele produces a sufficient amount of the specific proteins. Thus, a recessively inherited disorder shows up only in the homozygous individuals who inherit one recessive allele from each parent. We can symbolize the genotype of such individuals as E small, and the genotypes of individuals with the normal phenotype as either E capital or E capital E small, although phenotypically normal with regard to reduce regard to the disorder heterozygotes A capital A small. Many transmit the recessive allele to their offspring, and those are called carriers. Most people who have recessive disorders are born to parents who are carriers of the disorder but themselves have a normal phenotype. A meeting between two carriers corresponds to a Mendelian if one monohybrid cross A capital A small A plus A capital A small. The genotypic ratio for the offspring is 1 A capital A small, 2 A capital A small, 3 A small A small. Thus, each child has a quarter chance of inheriting a double dose of recessive allele and being affected by the disorder from the genotypic ratio. We also can see that out of the three offspring with the normal phenotype 1 A capital A small plus 2 A capital A small, two are predicted to be heterozygous carriers and 2 to 3 chance recessive. Homozygotes could also result from A capital A small plus A small A small plus A small meetings. But if the disorder or if the disorder is lethal before reproductive age or results in seriality, no A small individual will reproduce. Even if recessive homozygotes are able to reproduce such individuals, we will still account for a much smaller percentage of the population than heterozygous carriers. In general, a genetic disorder is not evenly distributed among all groups in humans. For example, the incidence of the Tay-Sachs disease is disproportionately high among Ashkenazic Jewish people whose ancestors lived in Central Europe. In that population, Tay-Sachs disease occurs in 1 out of the 3,600,000 deaths, about 100 times greater than the incidence among non-Jewish or Mediterranean Sephardic Jews, such as an even distribution results from the different genetic histories of the world's people during this technological times when populations were more geographical and hence genetically isolated. We will now examine the other recessively inherited diseases which are also more prevalent in some groups others.